All right. Hi, guys. So my name is Max. I'm one of the SIs for Dr. Maduro's 5A class. And today I'm just going to talk about a question that is on our SI worksheets. And we're just going to go over kind of PCR and like some of the questions that you might expect, right? And so ignore that little one up there. Not too sure why that came up, but anyway. Um, so the question is basically stating that, after we do PCR, we're trying to visualize our DNA, right? The problem is we make a mistake, right? We realize that we added dideoxy ATP, right? So that's something to keep note of. And so what this means is there's no 2' or 3' OH groups on any of the adenine molecules, okay? And so that's pretty much asking us like, okay, if this was the strand of DNA that we were given, what would that mean in terms of like the gel, right? So if this was our primer here in blue, and underlined is the gene that we were trying to amplify. What's going to happen to our gel, right? So remember that in ribose, we have our molecules, right? Each of these, has its own OH and whatnot. Remember that in deoxyribose, right? So the deoxy refers to removing the 2' OH, right? So in deoxyribose, we would still have our 3' OH, right? And let me number the carbons just so we're on the right track here. We have 1, 2, 3, 4. Then our 5th one actually is going to be over here, right? We have our little CH2OH thing, right? And so that's our 5th carbon right there. So this is deoxyribose, right? So we know that the phosphate groups, if we had them, they would add on to here, right? Phosphate would add on to this OH and this OH too, right? This goes with the 5', the 3' thing. So this would attach itself to a phosphate as well. So this creates the backbone, right, of DNA. So this 3' OH, the thing that you guys should be thinking about is this 3' OH is kind of integral to actually elongating the primer, right? So if I don't have that 3' OH, I can't really elongate my strand. And so that's kind of what it's telling us. We have dideoxy, don't mind my handwriting, it's with a mouse. Dideoxy meaning we don't have any of the 2' or 3' OHs, right? So that means that 2 and 3, we pretty much don't have anything to build off of, right? We can still make our phosphate groups, right? The phosphate groups can definitely still add to carbon 5. The problem is the phosphate group can't add to a hydrogen because it needs that oxygen, right? That's kind of what the phosphodiester bond looks like, right? It's the oxygen phosphate and then the whole phosphate group. So without our 2' or our 3' OH rather, what we want to think about now is what does this mean, right? What happens is without the 3' OH, we are unable to elongate the strand and we will stop elongation, right? So remember that in PCR we have the 3 steps. This isn't really going to affect the denaturation part because that's just heating. It's not going to affect the primer annealing, right? Because that's just sticking the primer onto the DNA. The issue is with the elongation. And so what ends up happening, and the reason why the adenine is important, is because if we think about our strand here, right? So I'm going to write out the strand that we would get as a result of our synthesis, okay? And so if we start from the primer, GCTG, right? I'm gonna put that as our primer, GCTG. I'm just gonna put a space between it, but if we were to add on our nucleotides, we know that T is complementary to A, right? And so what would happen is, okay, we stick our A, right? We have our A, our adenine nucleotide on there. The thing is, if our adenine nucleotide looked like this dideoxy on the right side, what would that do for the rest of it? Could we stick on a guanine, or sorry, a cytosine to complement that? And the answer there would be no, right? And so technically, our DNA strand would look something like this, where we have an extremely short strand where we only added the adenine because the I guess you could say like the guanine is not able, the cytosine is unable to add on to the molecule without the OH, right? And so now we know, okay, we have a shorter strand. What does that mean for our gel electrophoresis? And so for the gels, remember that in the gels, we have kind of different scenarios based on what happens, right? And so we know that longer strands will travel a shorter distance. We know that shorter strands will travel a longer distance. It's not shorter, just shorter. All right. So we know those two things, and our current situation puts us into the latter, right? Into the shorter strands category. Oops, let me. Our situation puts us right here, shorter strands, right? And so we know that, so let's just look at each answer choice. Number two, the strands would travel the same distance, so we know that wouldn't be the case, right? If we had a normal strand, we would do this entire thing where we have A, C, G, A, T, C, T, right? That whole strand that would be 1, 2, 3, 4, 5, 6, 7 additional nucleotides to the primer. However, we only have one, and we know that they will not travel the same distance. The second answer choice, the strands would travel a shorter distance, right? And does that kind of apply to us, right? If it's a longer strand, we know that you can think of it as it's being heavier. So if you're applying the same amount of force, so let's say you have two like rocks, right? You have one rock that's bigger, one rock that's smaller. If you apply the same force to the large rock, it's going to go like X amount of feet. Let's say it goes 1 foot forward. But if you apply the same amount of force to that small rock and you push it forward, it's going to go a lot more. It's going to go a lot further. Think of like kicking a soccer ball versus a tennis ball, right? If you're kicking a soccer ball with like an X amount of force, it'll go a certain distance, but the tennis ball is so much smaller, right? And so it'll travel a lot further. And so we know that it's not going to travel a shorter distance because it's a smaller strand. And we kind of figured out that since it's a shorter strand, it's going to travel a longer distance, and so that would be our best answer choice. We definitely have enough information to answer this question, right? We know that the DATPs will kind of stop our or kind of not transcription or translation, but it'll stop the replication right here, right? And so our entire strand kind of, you can think of as just this length, as opposed to the ideal length that would be all the way up to these A over here, right? So we know that for a fact, and we also know things about PCR and gel electrophoresis that would allow us to answer this question, even though those facts are not necessarily specified. So if you guys want more questions like these, go ahead and go to our SI sessions. Those times are posted in Dr. Maduro's iLearn page, as well as just look at our worksheets. You know, these are also linked on the iLearn page for 5A, and have a good day.